sharing my okay, people can see it, right?
Yeah, it looks great.
Yeah, okay, great. Well, first of all, I want to thank Hong, Jay, and Simo for putting together such exciting program for today, and also for inviting me to give the opening talk, even though I'm totally not a morning person, so hopefully I just speak just fine. Yeah, okay. So it's really a great pleasure to share some of our recent work with you all here. And yeah, I'm a developmental biologist. So one of the major questions we study in the lab is to understand, how the cell, like we are all sitting here and we are all coming from one cell, a fertilized egg, right? During development, this cell will divide, followed by differentiation, and become many different cell types. In the human body, we have more than 200 different cell types, but they are all coming from the one cell, and most of them carry the same genetic information, the DNA sequences. So how would these cells carrying the same genetic information end up becoming cells with different morphologies, carrying out different functions? That is still a big mystery in developmental biology. So many years of work, actually very well explained, like some very fundamental things in biology, such as DNA replication followed by cell division. So DNA replication followed by mitosis very well explained how one cell upon division will become two identical daughter cells. However, the question we ask as a developmental biologist is how cells become different during development. So of course, this is a big mystery. However, many years of work contribute to our basic understanding. For example, extrinsic cues such as signaling pathways, as well as intrinsic factors such as RNA molecules, organelles. This information can affect the cell fate decision to allow the cells post-division to become distinct cell fates. However, we are asking whether it's possible during this cell division, something can be internally inherited or segregated in a non-equal manner to give rise to daughter cells with distinct cell fates. So we know in developmental biology that not all cell divisions are equal. In another word, not all cell divisions give rise to two identical daughter cells. So there is this so-called asymmetric cell division, which is often carried out or used by stem cells, to give rise to two distinct daughter cells. So in another word, upon asymmetric cell division of stem cells, one daughter cell will remain as a stem cell. So this process is called self-renewal. And another daughter cell will take a distinct cell fate to go ahead and differentiate to become different cell types. So this is very often happening in tissues such as skin, intestine, blood, as well as in the male germ. So on the other hand, we are well aware of that not genetic information define everything about the what the cells carries. So it's very important that epigenetic mechanism regulate differential gene expression. In particular, the post-translation modification of histone tails play important roles to define a local gene to be either expressed or silenced to give rise to the unique gene expression pattern for each cell type in order to define this distinct cell fates. So this phenomenon is also very well recognized. So when we started this project, we really wonder whether these two phenomena could be connected. In another word, could the epigenetic information be inherited in an asymmetric manner during stem cell asymmetric cell division? So that's our starting question, but we need an organism or a system to in order to address this question. So first of all, we need a system to visualize histone information. So we designed a tag switching system, which is having two different tagged histones, and the upstream sequence is flanked by DNA recombination site, which can be recognized by the DNA recombinase induced by heat shock promoter. So using this very straightforward manipulation, we can distinguish these two populations upon a very straightforward manipulation, which is a heat shock. That before heat shock, we have this green fluorescence tagged histone, and after heat shock, we get rid of this GFP tagged histone sequence, so we can turn on the downstream red fluorescent tagged histones. So in another word, it's a straightforward manipulation, but can distinguish, the old proteins in this manner, this context histones, and the new protein, which is a new histones upon heat shock manipulation. So placing this molecular genetics design in the context of the cell cycle. So we started this experiment by looking at Drosophila male germline, which has a pretty long cell cycle. And in particular, they have an elongated G2 phase. So when we perform this experiment, which is a heat shock manipulation, so most of the time we hit the cells staying at the G2 phase. So this G2 phase germline stem cell, in response to the heat shock induction, will change the expression from green fluorescence tagged histone to red fluorescent tagged histone. So we allow this germline stem cell to rest until complete one cell cycle, so that the new histones can be efficiently incorporated during the subsequent DNA replication. So therefore, the germline stem cell will become yellowish, carrying both old green histone and red new histone. So when this germline stem cell under the subsequent asymmetric cell division, we found something really striking. We find as early as prophase, when germline stem cell, the chromosome in the germline stem cell under condensation, we can already see the domains carrying old green histone or red new histone. And upon sister chromatid segregation in anaphase and telophase, we can clearly see the separation between old histone and new histone. And in particular, the old histone enriched sister chromatids are segregated toward the future stem cell site, indicated by this asterisk. And the sister chromatids enriched with new histone are enriched toward the future differentiating daughter cell site. So once this observation was made almost a decade ago, and from that point on, we started our journey to try to understand, first of all, what are the molecular and cellular mechanism underneath this striking phenomenon, and second of all, how general this phenomenon is. Does it happen in other stem cell lineages, and does it happen in other developmental stages? So I hope to use the rest time to share with you some recent work towarding these goals. So first of all, when we first observed this asymmetric histone inheritance, we wondered, whether this is a beautiful artifact or is something really biologically meaningful. So we did the controls in the same tissue. We have stem cell undergoing asymmetric cell division, but we also have a lot of progenitor germ cells undergoing symmetric cell division. So we compared the histone inheritance pattern in two different stages of cells within the same tissue. And I hope you can appreciate this asymmetric histone inheritance is specific for asymmetric dividing stem cell. And this is just quantification. And furthermore, we also wondered, whether this asymmetry has any molecular specificity. So we use the same design to tag all canonical histones. So we found that both histone H3 and H4, which form a tetramer structure, both display this asymmetric inheritance pattern, but not for the H2A nor H2B, which are formed dimers. So through this experiment, we conclude is this tetramer forming H3, H4, which also carry the most post-translational modifications, display this asymmetric inheritance during stem cell asymmetric cell division. And recently, we also find not only old and new histone display this asymmetry, but the entire histone nucleosome density display asymmetric inheritance manner. So here we look at the total amount of H4. As you know, among all different histone, canonical histones, H4 is the only one which does not have any other variant. So the total H4 amount should be a good indicator for the nucleosome density. So we look at H4 inheritance, the total H4 inheritance, even without distinguish old from new, doing stem cell asymmetric cell division as a control in progenitor non-stem cell symmetric cell division. And I hope you can appreciate the H4 display asymmetry during stem cell division, but not in non-stem cell symmetric division. So through these old and new observations, we made a model to explain this phenomenon. So first, we propose the establishment of this asymmetry is built up during DNA replication. In another word, replication not only duplicate genome, but also deposit or segregate asymmetric epigenetic information asymmetrically. So one sister chromatase carry more nucleosome and also more old histone, while another sister chromatase carry less nucleosome and also enriched with new histone. Well, histone, I mean here is H3 and H4. And second, we propose that during mitosis, during asymmetric cell division, the mitotic machinery can distinguish these genetically identical but epigenetically different sister chromatids in order to segregate them in a biased manner. And third of all, we propose that in the future daughter cells, each of the daughter cells inherit different epigenetic information, will allow them to execute different functions. So I'm going to go through each of these steps briefly in the next couple of minutes. So first of all, during replication, we propose that there is a strand-specific incorporation between old and new histone, plus there is a biased fork movement. So together, these two events, we think, contribute to this differential histone deposition. Because of the time constraint, I won't go through all the details, and also because it's published, I just only want to show one of the striking figure or feature we have identified using the chromatin fiber to directly visualize old versus new histone distribution at the replication fork. So here what you see is one replication bubble. I hope you can appreciate the old histone is aligned on one side and the new histone is almost asymmetrically on the other side of the duplicating DNA. So yeah, so this is we'll keep understanding how replication contributes to this very striking asymmetric distribution between old and new histone during replication. So second step, we propose that mitotic machinery can distinguish the epigenetic distinct sister chromatids for their biased sister segregation. And this is regulated by both asymmetric centromeres between sister chromatids and the polarized mitotic machinery. So again, because of time strain, I just want to show you one image of that, where we find the microtubules in these asymmetric dividing cells is very polarized. I hope you can appreciate between these two centrosomes, the microtubules coming from one centrosome is way more than the other during the first half of the mitosis. And this allows this polarized attachment and segregation. And third, we propose that upon division, the two daughter cells inherit different epigenetic information, and this will allow these cells to carry on, have different affinity to bind to key components such as replication regulators, as well as transcription regulators, to allow them to carry out different functions. So here again, I just want to show you one critical image where we find during asymmetric cell division, the daughter cells, which carrying the sister chromatids with less nucleosome, probably having a more open chromatin feature. And this side actually has more affinity to bind to replication initiation factors such as CDC6. I hope you can appreciate in these asymmetric dividing stem cells, CDC6 has more binding on the sister chromatids toward the differentiating daughter cell side compared to stem cell side. And this asymmetric inheritance of this factor allow the daughter cells to enter the next S phase prior to the stem cell. So together, we our previous and recent work contributed to our understanding of these three steps that establishing, segregating, and reading out this distinct epigenetic information between the two daughter cells. So in the next couple of minutes, I just want to share with you some new data we have, trying to understand how general this phenomenon is. Does this phenomenon happen in other stem cell systems, and does it happen during development? So first of all, we look at another adult stem cell lineages in Drosophila, which is intestine stem cells residing in the midgut of flies. So in the fly midgut, the intestine stem cells give rise to the entire lineage. And not only that, but also these stem cells are the only mitotic dividing cells in the whole lineage. Therefore, if we track the mitotic dividing cells, we know there must be stem cells. So during this intestine stem cell asymmetric cell division, we use a similar dual color labeled histone, and I hope you can appreciate that it displays a phenomenon very much like the germline stem cell, where old and new histone are segregated for and asymmetrically get segregated during intestine stem cell asymmetric cell division. And this is just quantification. And finally, we want to understand whether this phenomenon also happened in mammalian system. So we have a collaboration with Dr. Hai-Chin Zhao's lab in my department, and Hai-Chin's lab studied the olfactory neuronal system where they reside these horizontal basal cells. So these are quiescent stem cells. Normally they're not dividing, but upon injury, they can re-enter mitosis and divide. So the postdoc in the lab, Bin Mark, who is also at the meeting, and he labeled these dividing horizontal basal cells using this keratin 14 as a marker. And he also tracked the histone H4 inheritance pattern during this cell division, and by also using the P63 as a transcription factor to label the horizontal basal cell. So here, I hope you can appreciate doing, this is an anaphase horizontal basal cell, and I hope you can see better here that during asymmetric cell division, the P63 enriched the site, which is going to become a future stem cell, carrying more histone H4 compared to the daughter site. So indicating this asymmetry could also be happening in mammalian system. So yeah, I just wanted to, we start building up a lot of collaborations with other labs. And I just want to give you a summary of this different degree of asymmetry between old and new histone, as well as this new phenomenon we found for between the nucleosome density, and we can observe in both fly system and in the mice system across different lineages. So with that, we are hoping to keep exploring the generality among different systems across species. And we are also wondering whether this asymmetry can be used as a potential new method to identify stem cells at a single cell resolution in vivo. So with that, I wanted to conclude my talk. And I want to thank everyone, who contribute to this work. And also, we actually actively collaborating with Jay Shaw with the chromatin fiber work, and also KG's lab for doing the genomics work. I also want to highlight Bin is here at the meeting, and he will give a poster later today. Thank you, and I'm happy to take questions.
Thank you, Xin. It's always a very nice talk. Thank you.
Thank you.
Very elegant work. So I think I have one question in the chat box. I have a lot of questions, but I don't have time to ask. So one question in the chat box we always take. He's asking, how were the new and old histone protein labeled differently for super resolution imaging?
Or how are they labeled? So in the super resolution images, we are just using antibodies against their tag, either GFP or M-cherry. Does that answer the question? Yeah. We also actually, on the chromatin fiber, we also use antibodies against old histone or new histone enriched histone modifications, such as H3K27 trimethylation. We use it as a old histone enriched mark, and also H3K14 acetylation. We use it as a new histone enriched histone modification mark.
Yeah, that's great. And I don't know if you want to just say something about these are commercial antibody or something unique in your lab?
These are commercially available antibodies. Okay. Yeah, but we test their specificity by in vivo.
Okay. And the other question.
Very nice talk. Sorry, a quick question. How do you propose to use this approach to identify new stem cells in different organs?
Oh yeah, so yeah, this is actually something we are wondering. Okay, this is still at early stage. So we found in quite a few systems, the stem cells are featured by inheriting, even just looking at the entire amount of H3 or H4 during cell division, especially if you want to look at anaphase and telophase, you can already start to see the difference between the two sister chromatid sites, even without labeling them with different colors. Yeah. So we wonder, because H4 doesn't have any other variant, so we think it's a very good indicator of the entire nucleosome density. So we wonder whether this is a common feature between stem cell division, like these two daughter cells do not have the same overall epigenome architecture. So one side is more open, another side is more closed. At least, likely, yeah. So we are still exploring. Yeah.
Yeah, that's great.
Yeah,
So,
this
I
is
think
very
that-
straightforward. So anyone can take a quick look. Yeah.
I think Yi-Hong has a question, and I can just read for him. Why all the histone all gets deposited to the same chromosome? Is the tail of histone required?
Yeah, so that's a very good question. So I didn't go into details. So first of all, it's not all or nothing. You know, it's not like all old histone go one side, all new histone go to the other side. Even by eyes, it's look quite striking, right? So it's asymmetric, but it's not all or nothing. And another thing, I didn't have time to go through in the details. So we find the old histone are more incorporated by the leading strand, and the new histones are more incorporated by the lagging strand during the replication. And we also find among different replication forks, there is a coordination for the fork movement. So we think the coordination between the strand-specific incorporation between old and new histone, plus the bias the fork movement, you know, toward the unidirection fork movement, together contribute to this bias distribution between old and new between sisters. But it's not all or nothing phenomenon, just to clarify. Yeah.
Okay, I'm sure that there are many other questions that we can't just go on. And
yeah, I'll be in the speaker.
Yeah,
I'm looking forward to talking more with you.
Yeah, she will be in the break room, and if you have a question, go ahead to ask her, or during the lunchtime. Yeah, same time. Thank you very much.
Sure thing.
Thank you.
Thank you very much.
Okay, our next speaker.